What?
We still have a couple of minutes.
That's fine. Yeah. Yeah, no, spring I've got invited talks, and then I think my first conference is Hawaii, which I was like,
That's nice.
yeah, a trip to Hawaii, yeah, that sounds really good.
I'm going DC January, February, probably March as well. Colorado, February.
It
Yes.
looks like they're ready to go.
Yeah.
I started the live stream. I just have to go back there and transition the slide off, and then
Okay,
when
perfect.
you're ready.
Yeah.
So we'll go 10 seconds if you want.
Oh, no, I'll do 20.
Yeah, I'm Irvine, St. Louis, where alma mater, where I got my fancy award from, and all my friends, family. I don't think anywhere in March, and then my favorite one is I'm doing the diversity symposium at Indiana, but it's my friend who's hosting it, so they just give you $5,000, and then they're just like, show up for 3 hours. But he's my friend, so he's like, will you stay? I was like, just stay the whole weekend. Yeah, of course. But yeah, it's so funny. Yeah.
Okay, good evening, everyone. I hope you can hear me, and welcome to Café Scientifique today for a talk on 3D printing human tissues. And before I start introducing our speaker today, I guess I should introduce myself as well. My name is Magdalena Bazalova-Carter, and I'm in the Department of Physics and Astronomy. And it's my pleasure to be hosting today's Café Scientifique. And it'll be my pleasure to host Café Scientifique next week as well. So just a reminder that there will be one next week on November 16th. And if you're interested in learning about how the Victoria Hand Project is helping people in developing countries and in specifically hand amputees, then you can come and learn about that next week. So at this point, I would like to introduce our speaker today. It's my great pleasure to introduce Dr. Stephanie Willerth. And Stephanie is a full professor and Canada researcher in biomedical engineering at the University of Victoria. She's also a co-founder and CEO of Axolotl Biosciences, right? And her research interests include tissue engineering and regenerative medicine. And Dr. Willerth has been very successful in securing funding and also in teaching. So if you can think of an award that a researcher or a teacher could be getting, it's probably on her shelf at home. And just out of so many awards, I would like to mention two that are from this year. And the first one will be her induction into the Royal Society of Canada as a member. So it's happened quite recently, so congratulations.
Thank you.
And the other one is her 2021 Teaching Award of Excellence, given by the Engineers and Geoscientists, British Columbia. And with that, Stephanie, the floor is yours.
Thank you so much. Let me see if I can get this to work. Thank you for that lovely introduction. I'm told I need to move the mic stand, so this film's better. Yeah. So it's been quite a year, and probably quite a year for everyone with COVID, but it's good to be back doing science communication in person. So I guess I'll start off by telling you guys a bit about how I ended up here at the University of Victoria, and then a bit about the work we've been doing 3D printing neural tissues, that resemble those found in your brain and spinal cord. And also a bit about how I founded my company and where all that's going. And just let me know if you guys have questions at any time. It's so nice to have an audience of real people and not speaking into a Zoom monitor, which I did lots of over this past year. So yeah, I guess if we want to start, which is actually kind of interesting, as I was telling Magdalena, I had recently had to go back home. I'm originally from a small town in Missouri called Lee Summit. And when I was growing up, I guess now famously or infamously, when I was 5 years old, I asked for a chemistry set and some tickets to an American football game, which still are probably the same things I would ask for Christmas, given now that I have a bigger chemistry set, which is my lab. And my current students do always ask if I still have the chemistry set, which no, and God bless my parents, because as a 5-year-old with a chemistry set, I immediately mixed together everything it said, do not mix these two things together, in our backyard, and had some pretty exciting explosions and made some interesting purple crystals back in the day. Again, just because I was recently back home, I now have my parents dug up like all of my 7th grade science fair things. My senior thesis in my high school was actually based around stem cells, which is shows exactly how long this obsession of mine has been going on. To date myself a little bit, in high school was when they cloned Dolly the sheep, and I thought this was like the most fascinating thing on earth, the fact that you could replicate another animal using somatic cell nuclear transfer. And so that's what I did my senior thesis on. I was one of the top students at my high school, which was a very large high school, which wasn't actually known for producing many things intellectual, which I don't know if people usually associate that with Missouri anyway. But I did end up going to MIT in Boston, where I was a double major in biology and chemical engineering, and that's where I learned about biomedical engineering and bioengineering. And I worked in a research lab where I isolated heparin from shrimp as part of their tissue engineering strategies. And I loved working in lab, and I guess it probably goes along with winning the teaching award. I also was everyone's tutor and TA, so I TA'd for the biology office. And I knew I really wanted to go to graduate school in biomedical engineering. And again, I had this big interest in stem cells. So I went to Washington University in St. Louis, where I pursued a PhD in biomedical engineering under the direction of Dr. Shelley Sakayama-Elbert. And a lot of the work we did at the time was we developed a protein-based biomaterial called fibrin, and we engineered it in a way to make neural tissue from, at the time, mouse embryonic stem cells. And it was a really exciting time in the stem cell field. The interesting thing about the pluripotent stem cells I'll talk a lot about, which actually I have props. So now that we're back in person, like one of the greatest tragedies, of course, was when you had to do Zoom things. So I'd have to bring my stuffed animals home to talk about them on the Zoom. So you have stem cells, like the one I have here. And the type of stem cells I worked with, embryonic stem cells, are considered pluripotent, which means they can become any cell type found in your body. I don't have the whole range of cell types here, but if you come to my lab, you can see a whole bunch of them. A lot of people get very cheered up by the fat cell. We try to make neurons in my lab, which are here. Mouse embryonic stem cells were derived in 1981, and then in 1996 is when Jamie Thompson at Wisconsin-Madison, where I did my sabbatical, derived the human version. And it's really interesting to go back and study the history at the time, because Jamie, when he derived those cells, it was really controversial because they were derived from embryos. A lot of the political movements would tie that to abortions and things like that. So it was very controversial. He had to wear like a bulletproof vest when he was doing this work. And because the US government didn't fund it, it was actually funded by the organization called WARF, which is sort of the tech transfer office at Wisconsin. So 96, that was like the brand new technology. So when I was in graduate school, which was 2003 to 2008, there was a huge interest in how do we turn these embryonic stem cells into these cell types, especially into functional tissues you could use to replace diseased and damaged tissue. And so at the time, again, working in neural, we had a big interest in using stem cells to treat spinal cord injury. And so one of the first clinical applications of these embryonic stem cells was actually deriving a cell called an oligodendrocyte to be transplanted into the injured spinal cord. So I was quite excited at the time because it seemed like for these neurological diseases and disorders, these were getting therapies into clinic. And so it was just a really, really exciting time to be working in the field. And additionally, while I was in graduate school in 2006, I feel like now I'm giving history lessons, which makes me feel very old. 2006, I went to the International Stem Cell Society meeting in, someone's got their phone ringing, in Toronto. And I was a grad student at the time, and my boss and I, we go to the plenaries. And at the time, Yamanaka gave a talk about how he treated these essentially mouse skin cells with different factors. And he was able to turn those cells back into being stem cell-like cells. And I remember commenting to my boss at the time, I was like, well, if this was real, this is going to be like the biggest discovery ever. And those were the discovery of induced pluripotent stem cells, and Yamanaka won the Nobel Prize a short 6 years later for it. So it was, in fact, a really, really huge deal. And this was very exciting to me for a number of reasons. One, I was doing my PhD in Missouri at the time, so a red state, so there was like controversy over using embryonic stem cells. From a purely selfish point of view, there was one embryo that made me, and it's done. I'm a human, so I'm never going to have my own personal embryonic stem cell lines. But I can definitely give my skin cells to make induced pluripotent stem cells. And in fact, in our research lab, now at the University of Victoria, and with our collaborators at UBC, that's exactly what they do. And so you can actually get, and this is what Hakan Nygaard does with his patients. They do a blood draw, and then in this reprogramming laboratory, they treat these blood cells with factors, and they go back to being a stem cell. And then we differentiate them. We actually bioprint them into neural tissues. And the patient cells that we've gotten from his Alzheimer's patients, those tissues actually exhibit Alzheimer's symptoms in a dish, so we can actually track them. And so the next step for us is to actually screen drugs on these 3D bioprinted tissues. So yeah, I think it's really cool. I think that this is where a lot of the personalized medicine is going. Being able to have these patient samples and culturing them and then screening the drugs specifically on patient-derived, either pluripotent stem cells that are derived into neural tissues, or also we recently just got in some cancer glioblastoma patient cell lines from my friend at UCLA. And again, we're going to use these for some drug screening. So that got a little bit ahead of myself. But anyway, that's sort of the stem cell portion of our work. So yeah, my graduate studies, I spent a lot of time optimizing this protein material called fibrin. When you cut yourself into blood clot forms, that's fibrin in action. So I made a really good version of that for culturing the mouse cells. I then went and spent 2 years at Berkeley, which for those of you who might have seen some of the things with COVID last summer, ironically, my postdoc, I spent studying the evolution of HIV and characterizing it. So then when COVID hit last year, 2020, I got recruited back into some of the COVID projects because I was one of the few people on the island who'd actually worked with level 2+ containment viruses and things. So I got to consult on a lot of those things. Yeah, if there's a good thing to be, it's a biomedical engineer in a pandemic because everyone will need you. So yeah, I took a brief detour to do my postdoc in California because my husband had a job there as well, and we had lived in California when we graduated college, and then he moved to St. Louis while I did my PhD, and then he wanted to live in California before he made the final stop, or not the final stop on our journey, but present day stop, which is me getting a faculty position at the University of Victoria. So, and that was now 11 years ago. So UVic flew me up. They were looking for a biomedical engineer. Apparently everyone else they interviewed did bone tissue engineering, and I said, well, that's really boring because it's pretty simple. I was like, when you have neural tissue, you've got these electrical signals, you've got these chemical signals, you've got all these cell types. It's really interesting. And I was like, also, your medical school at the Island Medical Program does all neuroscience. And the dean at the time, Oscar Casario, I met with, and he thought I was great. So he was like, hey, if you come here, you can have your lab in our building. And my lab's been in the building ever since. And it's still a really lovely lab. If you ever want to stop by, you can come see all the places where we culture our stem cells, as well as our bioprinting collection. So that's how we ended up in Victoria. Oh, yeah, I guess the other part of the story I'm supposed to tell is like, everyone's like, why Victoria? And I was like, so we were in Berkeley, or we were in Northern California at the time. I was working at Berkeley. We were inland. So I interviewed at Alabama, and a school in upstate New York, and Victoria. So, and for those who remember the time back then, the Canadian dollar was actually stronger than the US dollar. So I think we are the only people probably in the history of whatever to move to Canada because the Canadian dollar was worth more, and we actually paid less in taxes compared to what we paid in California. So that's how we ended up here. I wrote a bunch of grants and had a research lab. And so I was like, okay, our lab is going to focus entirely on engineering neural tissue from these human-induced pluripotent stem cells. And so that's what we really focused on during my first few years. At UVic, we established some really nice collaborations with people from mathematics and mechanical engineering. And then it really sort of got interesting in, so I think it was 2016, a company called Aspect Biosystems. They spun out of the University of British Columbia. Conrad Wallace was one of the founders, along with Tamer Mohammed and Simon Bayer, who were his graduate students, and they had made a bioprinter. And so this was interesting to us because how we used to make our 3D tissues is you would have a pipette man, which I do not have, but if you come to my lab, so you have a pipette man, you would add your different components of your you would add your fibrin, you would add the components that would essentially induce it to clot, you would seed your stem cells in it, and then you'd add another layer of fibrin to make your little tissues. And so you're just manually making these, and you'd usually need to make about 24 of them to get enough cells to analyze and see what your tissues were looking like. And so the process would take you 4 or 5 hours. And you'd have your plate of cells and then manually change media and things like that. So when we saw the bioprinters, it was like, okay, we could automate this process and potentially make it more reproducible and produce more of our tissues for analysis. And so when on the good days when our bioprinters are working, that's exactly how that goes. Sometimes it's not as smooth. But anyway, so Aspect approached us and they're like, so we have this really cool bioprinting technology. They had spun out of an electrical engineering lab. And they were looking for people with material science expertise, which is what we had. So they're like, could you work on making some bioinks that are compatible with our printing system? And I'll try and pass this around. We have a prop. So the interesting thing about the Aspect Biosystem printer is they use these microfluidic printheads, which was a really new approach to bioprinting at the time. A lot of the bioprinters are similar to what you see with traditional 3D printers where they're just extrusion. Think about the melted plastic filament that makes little shapes. I don't think I brought one, but my lab 3D prints a lot of plastic things in addition to all the tissues we bioprint. And so with bioprinting, where it gets a bit different is you have to use a special bioink that'll keep your cells alive. And so usually you can't process them at those high temperatures because it'll kill your cells and kill the properties of your ink. And the nice thing about these microfluidic printheads that Aspect makes in their system is it gives you really good control over the flow. So it's very gentle on your cells and your ink. So it really allows you to print tissues and keep the cells alive at the same time. So if you guys want, I'll just pass this around through the crowd. It's a printhead. So we actually have the big printing machine, and then you actually put them on the machine as you print your different tissues. And so usually on a good day, you'll use one printhead to print a couple, probably like 150 tissue models. And yes, it takes the students a long time to get enough cells to print them. Yeah, so it's made from a plastic material, and they have different channels. They've got a few different configurations that allow you to print different arrangements and combinations of cells as well. So they had reached out to us about collaborating, and we got some grant money to do that. And we were actually the second installation of one of their Aspect RX1 printers in the world, which was really exciting. And it took a long time because when you print cells, you have to use these sterile tissue culture hoods. So it took a long time to get our hood installed. So it was Valentine's Day 2018 when we actually got our bioprinter. So it was quite romantic. I mean, we've been waiting for it for a year, and so we were so excited to have it installed. And so then our lab just went crazy, printing like every single cell type possible. And so it was really exciting. So we had a lot of these human-induced pluripotent stem cell-derived neurons, which at the time, people were printing on these other systems that were harsher on cells. So when they would print their stem cells, only about 50% of them would live. And then again, just sort of the nature of the field, because it was from people who were from mainly mechanical or electrical backgrounds, they would culture these tissues for 3 days and be like, okay, 50% of the cells are alive. They express some neural markers, it's been 3 days, and call it a day. And that's really not mimicking what the tissues in your human body do. And we knew that because in our previous work, before we would bioprint things, we'd keep our tissues in culture for, 30 days to 60 days. And then they would actually mature, and they would start to secrete the chemicals you would see in the brain. They would also send electrical signals like you would see in brain tissue. So, so that was sort of where we came in, as we had this unique bioink that we had made and formulated on the Aspect system. A team of sisters, Emily and Lila Abelseth, who also have both worked at our spin-off company, Axolotl, were the ones who really drove that work forward, and it was published in the journal ACS Biomaterials Science and Engineering, and it was actually selected as one of the methods of the year in 2019, our bioink formulation. So it was quite exciting. And people were really excited by the fact we were able to get neural tissues that were like not only surviving, but we actually took them out in that paper. We cultured them for 46 days, which at the time was pretty much unheard of for someone to be able to culture these tissues for so long. But as I mentioned previously with the Alzheimer's work we do currently, being able to take them out this long is really beneficial for drug screening, because then you can actually monitor these Alzheimer's symptoms. And we're currently doing the same thing with some of our Parkinson's-derived human-induced pluripotent stem cells. So being able to track the symptoms over time and having these stable tissue models is really important if you want to do what I would consider clinically relevant drug screening. Magdalena's taking photos of me. So anyways, we got the printer in, and we started printing a bunch of different things. Laura de la Vega, who's one of the co-founders of my company, Dr. Laura de la Vega, she did her PhD with me, and she wanted to print miniature spinal cords. One of the first papers we published with the Aspect, she had printed these miniature spinal cords, and then she had showed that if you treat them with different chemicals, you would get different compositions of the different cell types found in the spinal cord, depending on the different concentrations that you used of these chemicals. And so this was kind of exciting, because it showed we could tune what was going on in our different tissues as well. And yeah, this work was all sort of interesting, because a whole bunch of work came out all at the same time. Like, we submitted all the papers around the same time, and they sort of came out in different orders. Yeah, so that was really exciting. So, and then Laura actually built upon that work and had a paper come out recently where instead of just including these small molecules in the media, we actually encapsulate them inside of these small spherical particles, which in previous times I used to have like big posters I would bring up and be like, here's what they look like in SEM. But yeah, you can Google SEM, scanning electron microscopy images of microspheres. So they're just like these small spherical particles that we make from a polymer called polycaprolactone. And we use emulsion in order to get these different drugs inside of these microspheres. And then we had previously characterized them, and we saw we got release over 40 to 60 days from these microspheres, which corresponds to when we essentially want our tissues to differentiate. So we had sort of done that, and then we started bioprinting, and then my students were like, well, we could combine these, because if we have these drug-releasing microspheres in the ink, it means we'll have to change the media on the cells less often, and they'll be more likely to mature into the neural tissues that we want. And so that was some of Laura's work that just came out in Advanced Nanobiomed, where she encapsulated 2 different types of these small molecules into our bioinks, and then she was able to use them to produce functional spinal cord tissues, which was really exciting. And that's in a paper in Advanced Nanobiomed Research, which if you're interested in looking at all of her pretty pictures and data, it is open access. Also at the same time that Laura was doing this, I have another amazing PhD student, Ruchi Sharma. And so she was looking at similar things, except instead of spinal cord, she was really interested in printing brain tissue to model Parkinson's. So she used these microspheres to deliver a drug called glucosterone. And glucosterone is it's derived from plants, but it's usually used as an anti-cancer treatment. But an unbiased screen found that it was the most effective small molecule at producing dopaminergic neurons from human-induced pluripotent stem cells. And so we had previously done some studies. Again, we were the first group to encapsulate it and generate controlled release, because usually people are just using it to treat cancer cells as opposed to tell cells to become functional brain tissue. And so we published that. And so Ruchi wanted to combine that into our ink to see what sort of brain tissue we could produce. And so that's what she did. It came out in Frontiers in Bioengineering and Biotechnology in 2020. And so her paper, where she printed these miniature brains, using our microsphere-laden bioink, we actually got neurons that express tyrosine hydroxylase, indicating they're dopaminergic neurons. So this seems like it could be a pretty good system for modeling Parkinson's disease. And the paper was really well received. It's only been out a year, and it's already been cited 43 times. And that technology is actually something that we've got our patent pending on, which is what we're using at my spin-off company to sell. And we've actually renamed that bioink. We call that bioink that has the drug-releasing microspheres to produce brain tissue, we call it BrainPrint. And And the base ink that we used in our lab to print the initial spinal cords, and we actually printed some cancer models with, we've called tissue print, because as we've started the company and we've gotten a lot of beta testers, it turns out our base ink that we had designed to grow neural tissue is actually really good at growing a lot of different types of tissue, not just neural. So we've expanded it back to make that one more generic. And then as we add in these different combinations of drug-releasing microspheres, we get tissue-specific versions of our ink. And we also we're just in talks today with one of the local cardiac surgeons about optimizing an ink for printing heart tissue, which we would call heartprint, because one of the big issues with potential drugs is cardiotoxicity. So, so let me think. So we got a bioprinter. We got our first bioprinter, the Aspect one, and then I have to track things. It gets weird when COVID hit. So COVID hit, everything sort of went crazy. You might have seen our lab and some of my colleagues in engineering. We ended up making a lot of face shields for Island Health, along with some other medical supplies. No, those were using different 3D printers, although some people thought that we were able to use our tissue printers to make face shields, but that's not actually the case. The tissue printers are way more expensive, and the ink is way more expensive. But we did do that. And interestingly, so at that time, as a professor, what happened, we had to shut down our labs as required by health and safety, province health orders. So we had frozen down all of our cells. We couldn't do any wet lab stem cell related work for 3 months there. And ironically, I received a large grant in that time to buy equipment, which was ironic, but it worked out because at the time everyone was, probably everyone's still freaking out about the economy. So when the world is totally gone upside down with all this stuff, and you tell a salesperson that you're like, I just got $150,000 to spend on equipment, you get a really good deal on a lot of equipment. So they ended up giving us we ended up buying a live cell imager, which takes photos in real time of our tissues, so you can monitor what the cells are doing. We can monitor how differentiated they are into neural tissue and things like that. And then they also sent us, so they're now called Bico, or for bioconvergence, but they were originally Cell Inc., which was the original company that sold BioInk and sold bioprinters. So we got one of their bioprinters called the BioX, and then they also sent us another printer called the LumenX, which is a really interesting tool for printing blood vessels. So when you think about the tissues in your body, even in your brain and skin, you've got blood vessels everywhere that's nourishing those cells. And so that's one of the things that's really hard to replicate. And so with bioprinting, one of the reasons we have sort of the two different printers is the LumenX is really good for printing vasculature or blood vessels, and you can actually print other tissues using either the Aspect system or the BioX system onto that vasculature in order to get a functional tissue construct. So during COVID, we got a bunch of different bioprinters and equipment to use. So one of the things that makes our lab really unique is that we have all these bioprinters, so we can actually test different bioinks side by side, because each printer has different nuances. It's probably, if you just even think about like your home printers or even just traditional 3D printers like Ultimakers versus Enders, each one has its own finicky different things. And so we've been able to try out a lot of stuff and compare performance side by side of our inks, of other people's inks on all of our different printers. So it's been kind of an interesting ride. And now that I think people have started to know our group, we actually get sent a lot of bioinks, which I think is really funny because they know I have a company and we have our own ink. But for the people who work in the field, each of the materials can be optimized for different applications. So I always don't think we're necessarily directly in competition with each other. But it's been interesting that all these companies have sent us a lot of free collagen and Gelma to print, and my lab likes it because we have a lot of interesting things to play with and study in our group. So I don't know. I think I'm telling a story of my life, and then I'm like, and then COVID happened, and then I can't put any events in any proper perspective. During the pandemic, we actually incorporated our company, Axolotl Biosciences, the week everything shut down, which I think in retrospect I don't recommend. But the story that I tell is we had been at a conference, and again, this is pre-COVID, so I'm always like, oh yeah, so we saw people in person, and I was talking to the CEO of Bico, or what at the time was Cell Inc., and he was talking about our ink and how well it worked and things like that. And I was like, oh yeah, well we have these grants to commercialize it, but Aspect only has got 10 of their printers out there. But, you know, we've got all this money. I'm bioprinting my tissues. Like, I'm a professor. It's awesome. You know, I get paid to give talks on my work everywhere. You know, we're at a conference. We're having fun. Life is good. And he's like, oh, but we would sell your ink through our company. And I was like, oh, well, that's different, because they have like thousands of printers worldwide. And their printers were cheaper too. So I was like, oh, well, like, yeah, you guys have like 3,000 printers. And like, you know, I was like, that's a huge market for us to sell to. So that's when we had decided. And my graduate student, Laura, she was very keen on starting a company. So we went and met with the Coast Capital people at UVic. We won the internal planet competition, so we got some money from that. We had gotten some funding from both INSERC, they have an Idea to Innovation grant program, and the Michael Smith Foundation for Health Research and the Pacific Parkinson's Institute also had given us a bunch of money to commercialize our bioink. And so it all seemed to be coming together. You know, we had all these companies that wanted to distribute our ink. We incorporated, we got some IRAP funding, and of course everything shut down. But the team was very hardy, so we did actually end up having 5 full-time employees in the summer. So that summer of COVID, I ended up having to hire everyone. So I had my research lab, who I was supporting, but couldn't go into my lab to work, and I was too sad to deal with it. So I was just like, okay, well, we're not gonna, some people were like, we're gonna just have the lab do computational stuff, and we did not. We just all moped a bit. I had the face shield project, so I was overseeing a group of 15 co-ops that were working on COVID-related projects, and I had my company of 5 people, which they actually did a lot of stuff to help get us started on like process mapping and development, and it was kind of interesting. So we had always worked remote because we incorporated in the pandemic, and so it was sort of interesting that we all had our team remote meetings, and then we were able to get back into the lab in July. And also on the company side, we do have a rental contract with UVic, so axolotl works out of my lab at UVic, and we just pay when we use it for company production. So then back in July, we were all able to start, you know, slightly going back to work, and we were able to recruit beta testers for our bioink at Axolotl, and it's gone really well now that we've finally started to get some results. So we've had Bico was testing it, Stem Cell Technologies, a company called Bioprosthetics out of the US, actually just got some really nice data showing that our ink can be used to print human bone tissue, and they're actually writing a paper on it. So I don't know, they get stars as one of the better beta testers if you just show up after we send you our product, and then you're like, okay, is it all right if we write a paper? Yeah, it's great. And then they're like, yeah. And they want to translate their work first into animals, but then into humans with our ink. And so it's kind of interesting with our bioink, because I come from a background of studying spinal cord injury. And so our bioink is one of the few inks that is in a formulation you could make compatible to make a tissue to put into humans. The versions we sell are just for academics to use for drug screening or printing tissues, but there is still that possibility. And we have talked to companies in the United States, and then obviously our beta tester Bioprosthetics, about what it would take to meet the manufacturing standards to take that ink for clinical trials. So there's actually a company called Huma Biologics out of Arizona, and they make a GMP version of their ink, which is made from a protein called collagen, and they literally just do extractions from cadavers. So it seems really wild, but anyway, we talked to them, and they were like, if we find people who would potentially want this, could we partner? And I was like, oh yeah, we can definitely partner to take things to clinic. So while our current version of our ink is really sort of the low-hanging fruit academics, tissue modeling for drug screening, there's always the longer pathway, and that's what our collaborators at Aspect are really focused on. And I know they've been presenting some really interesting preclinical data for treating diabetes and also liver disease, where, you know, they're taking their products and testing them in animals first with the idea to get the regulatory approval to eventually take them into humans. So that's sort of the like higher grade things. Also, again, in the field of 3D printing, which is just, I think, really awesome and really cool, and I think the community is pretty welcoming. One of my friends, Dr. Adam Feinberg, his company Fluidform has a similar process, but instead of using cells, they print a really dense version of their ink to make heart valves. And they're also moving towards taking those to clinical translation. And Dr. Jordan Miller out of Rice, who founded a company called Volumetric, which is when I was talking about the printer that we use to print blood vessels, that was his company's technology. They just sold to 3D Systems for $45 million. And they're expanding their footprint in Houston, and their valuation can go as high as 300 million if they hit their targets. So it's sort of a really crazy, interesting time in the 3D printing space and for people looking at these technologies. So yeah, I think that's a long way of saying we had a lot of beta testers, and then 3D printing is really cool. So yeah, so we were able to actually start shipping our ink out last summer. And one of the fun things about the field and the technology is it's really global. So we've got beta testers in the US. We collaborate with a Finnish company called Brinter. We've sent our ink to Spain, to RegMat, and they presented on our work. Stem Cell Technologies, I think, presented on our work in an Australian 3D printing conference, which so it's really interesting how global the market is, and we've learned a lot about shipping things around the world now. Cell Ink is in Sweden, so we've sent them a lot of ink there as well. And we do send a lot of our beta testers are local at UBC, so we've got people over there. And one of the coolest things, I think, is at UBC they also have one of the Aspect printers and one of the Cell Ink BioXs. And so the coolest thing is for us to send someone our ink and our file, and they culture their own cells, but they're usually neural cells similar to what we do. And then they can just print that structure there and just have their own, essentially recreate our tissues elsewhere. And so that's been really fun, is being able to send our designs in ink places and having them print the tissue models and use them sort of plug and play, which is, I think, a lot of fun. Also, pre-COVID, I used to have to go to Vancouver a lot. I'm assuming this will probably start up again at some point, but it hasn't yet. But it's been interesting to just be able to ship stuff over there and then just have students over there being able to print and analyze our technology. And we do have a team at UBC that's actually working on some of our 3D printing stuff for axolotl, and they're trying to figure out a way to predict whether or not a structure will print based on the design file. So yeah, it's been really exciting. So it's been cool seeing like what all of our beta testers have done. As I mentioned, like for example, the bone tissue stuff is really interesting to me because in my home lab, that's something we would never do. We're very neural focused. You know, we've got our Alzheimer's models, we've got our Parkinson's models, and we've got our cancer glioblastoma models. And so it's been really cool to see people print tissues that we wouldn't normally look at. Like our collaborators in Texas have been printing heart tissue with our ink. Our collaborators over at the Prostate Center in Vancouver, they've actually been able to print functional testy organoids that produce sperm. So, yeah, it's either very exciting or everyone's scared. So yeah, just being able to see all the different things that our beta testers have been able to do with our ink has been really, really cool. And yeah, as I mentioned, some of our ongoing work is, you know, modeling these things specifically from the patient-derived cells. I think where this all goes in the end is you're going to see 3D printing centers at your hospitals where you'll get a biopsy, or that you'll biopsy, or they'll take your blood, they'll make your stem cells, and they'll print a bunch of your tissues and then screen drugs and see which ones the tissues respond the best to, and then that'll be what they prescribe back to you, is definitely where I see the field going. Magdalena, how am I doing on time? Okay. Either way is fine. Does anybody have any questions? Oh, sure. Let's start here and then go over.
So where is approximately, in your opinion, the technology for actually printing like kidneys or something like that to take care of diseases?
Can you repeat the question, or should I?
Sure.
The question was, where do you think the technology in 3D printing full organs maybe is? Yep.
Well, you said kidneys, so I'll give you the spiel, which is like, I think they're pretty close. I know they've been in preclinical testing to do skin at the University of Toronto, but skin's pretty simple. Kidneys, I think, will be a little bit more difficult, and also just like getting them to test. Kidneys are more realistic because, you know, you do have a defined structure. It is a replaceable thing. Like, this is something that gets a bit tricky, like, as I was mentioning, Adam Feinberg from Carnegie Mellon and his company Fluidiform, like their eventual goal is to make replacement hearts. And I'm like, yeah, and I'm sure you will. This is where the part where I've like always like went to crawl under something because I know I'm just tempting fate is I feel like, so I'm always like, you would not bioprint a brain. And I think this is just like Elon Musk will go on Twitter and be like, I'm bioprinting brains, order yours today. Because if you look at the diseases and disorders of the brain, so one is, again, coming from spinal cord injury, traumatic brain injury. you know, you don't just like miss a part of your brain and then you're going to like scan it and print that in. The other thing is you get into some ethical issues, which even with our tissues are small, so it's not that, but there's ethicists out there who are like, how big should you make a human brain tissue, especially when they make the organoids that resemble the brain. It's a good party question to ask, because if you ask the Germans, they're like, no, these brain organoids aren't doing anything like remotely relevant. And then if you read some of the papers, they're like, we've detected brainwaves in them. I'm somewhere in the middle where I think we are replicating some key electrical and chemical signaling. I don't think there's any good way to know if you've replicated thought, especially in a tissue in a dish. But it comes up a lot, especially when they talk about like even combining multiple segments to make larger cellular organoids. So that's why I'm always like, I don't think you'll bioprint a brain because the ethicists will go crazy and replace that. But like for the things that are traditional, like lungs, heart, I'd probably say it's like 5 to 7 years. Although with the amount of money that's being poured into 3D printing, it's a $35 billion industry. And 3D systems that acquire volumetric, like they aren't necessarily a healthcare company, but they're putting that money in. I think it's United Therapeutics is also throwing a lot of money at it. So I could see some of this stuff advancing quite rapidly given that. Also, at the other area that's really interesting, which pre-COVID, I was talking to Health Canada, was actually getting some of these human tissue models into the drug development pathway, because we currently test them on mice and rats. But testing them on a human model is probably more relevant. And so we were talking about ways to get those into the regulatory pathway, and then the world ended with COVID. But then also we made vaccines in record time. So we'll see how this all shakes out. So that's kind of the spectrum. Like I think you'll probably see skin being bioprinted on people in like 2 to 3 years. We'll see where all the aspect stuff goes. I think that's 4 to 5. The other thing with those right now as it stands, 3D printed kidney tissue is both a medical device and a cell therapy, so it's got twice the regulation. So that's why I'm always like, if someone wants to partner with us to take our stuff to clinic, then yes, but we aren't going to, in my cute little lab, go through all these regulatory issues.
Okay, thanks. So I know there was another question, but I'm going to ask a follow-up question. So if you were about to 3D print a human kidney, how long would it take? It's not really relevant, but I just out of curiosity.
Yeah, I think it'd be pretty quick. Like it'd probably just take, even if it's the size of, like a normal kidney, it'd probably be an hour. Because like when we print our constructs, they're usually printed in like a minute. So yeah, the printing process is pretty rapid.
That's very quick. Thank you. Okay. Okay. Let's.
For that to happen, you'd have to have many inputs and print different things at the same time. Could you explain that process a little bit to us?
Yeah, so it depends on so with the aspect printer, you have the different print heads, so you could actually switch between your different cell types and put them in your specific places based on where it says in the file. That's partially why we have a UBC design group predicting on whether or not structures will print. It came about from the fact that currently the way we predict if a structure is going to print is we have cheap materials that we put food coloring in, and especially if we want to do like multiple cell types, you'll print the cheap materials to see that you're getting the things localized where you wanted, and then if it looks okay, then you go move on to using your really expensive cells to see that they're in your right locations. And so what we do when we print the more expensive multiple cellular constructs is we usually use fluorescence to label them so we know which cells are in the right locations, because I mean, the aspect system is amazing, and the printhead can localize things here or there, but you also, we were the second people to use it and the first people to publish. You have to actually show you're putting things where you say they are, because if you just look at cells, they all look sort of clear together. So, yeah. It would depend on which technique, but ours are currently linear, where you would just switch between what you'd want at that spot. Some of the light polymerization stuff, you could potentially get some more of the vasculature being polymerized simultaneously. So.
Thank you. Another question. Okay, on the right.
Printing various structures, neural or vasculature or whatever. Is the morphology all from the printing, or do the cells organize themselves as well?
So I'm going to repeat the question. Do the cells organize themselves, or is it based on your file that you give to your 3D printer?
It's both. So with our ink, it depends. Like you can change, just like when you 3D print, like how you do your infills. And so you can print stuff and your cells will sometimes align with your ink, especially if you're using like a core shell formulation where you have like a core material and an outer one. So then you can get alignment from that. Our neural cells do interact with each other and interact in 3D as they sort of remodel the ink and replace it with their own ECMs. I know people have printed like cardiac bundles and things like that as well.
It's very interesting. We have another question here. I'll go there.
So standard 3D printing, like FDM printers, that they often use G-code for printing. So in the industry, is there a standard language?
Yeah, no.
Okay, I'm going to repeat the question again. Is there a standard language for 3D printing of biomaterials?
No, we still have to slice. We still have to slice and make G-code. It's just simpler. So, although I would say we print simpler structures than what you do with traditional 3D printing. So it's not as complex. But as I was mentioning, there are things with the infill and infill percentage that you can play with that have a big effect on your cell behavior. But yeah, we do. Yeah. Yeah, it all gets sliced to that. Yeah.
Maybe you could do it some 3D. You could do some 3D printing as well. It sounds like you're familiar with G-code.
And I will point out that the collaborator is talking about the printed bone. They actually did modify like an ender to extrude our bioinks. So it is possible to make like the homemade versions. Like the Aspect's one of the high-end printers, and then the Celllinks are sort of the mid-range, although they actually are coming out with a clinical bioprinter. And then I just think it's really funny, like, because there's like a whole industry of how you home make a bioprinter. So
Interesting. I'll come back to you. I'll go in the back, white shirt. Thank you.
I have a background in nursing, so I know what Alzheimer's looks like in people. What does it look like in cells?
A very interesting question. What does Alzheimer look like in cells?
they're very disoriented. No, no. They actually, so you actually can track like your tau and amyloid beta levels, and so they do increase over time, just like you do detect. So you can see the tangles and the plaques, and so the fact that you can detect them is sort of what we use as the proxy. And that's what you would normally see. I mean, because you see it as it manifests in a patient, but technically you don't diagnose Alzheimer's till they die and you actually get the pathology. The other thing that I think is really interesting is now that we've got a couple of these different cell lines, is just seeing the patient differences and sort of how they grow and are cultured, because I think it's going to be one of those things where you find, a lot of different mutations that have the same phenotype, just because Alzheimer's is such a broad disease.
Thank you. There is a question from the gentleman there.
Back when you did the tissues manually, did the blood vessels just grow as opposed to now when you're 3D printing? And do I have it correct that you just change the head on the same printer to do the blood vessels when you're printing? And also, you said back in the day you had cells go for 46 days. And how long does the 3D printing last for?
Okay, the question is really about the differences of manual printing of tissues versus automatic printing of tissues, and what is the differences in blood vessel geometry, perhaps, or anatomy?
Yeah, so when back in the old school days, we only used neural cells, so we didn't have blood vessels present at all. Now what we do is we have two different ways. So there's a company out of California that makes these little blood vessel baskets, so you can just print your tissues around the little basket to get your blood vessel. We don't have blood, it's just that you get better cell culture media perfusion. And then with Luminex, you can actually customize what those blood vessels look like and then print on top of it as well. Was there another part to the question? Oh, yeah. So what happens is, what'll happen is the cells will remodel the ECM, but they'll stay in the structure. So like our miniature domes stay domes for 60 days. And so that's one of the things that's sort of nice, is that the cells stay in that structure. Because other studies, they would degrade the ink and then sort of become a 2D culture.
Is there a question here, or not anymore? The question is if there is any work done around diabetes.
Yes. In fact, Aspect's tissue that they've been developing with Tim Kiefer, who just took a position at ViaCyte, I think is their chief scientific officer. So that's actually one of the big indications. Canada has a very interesting position because of how cell therapy is being done here. A lot of the diabetes clinical trials are being done in Canada because there's a facility out of Alberta that I think does pig islet isolation, But because of this, a lot of the US companies are actually doing their diabetes clinical trials up here. And I know Tim Kiefer was leading some of the first ones using human embryonic stem cell derived cells for that. That was the first in the world of its kind. And then with Aspect, they were translating some of that technology to be a 3D printed tissue. And so they're evaluating that in animals currently with the idea of taking it to humans hopefully one day. And then obviously, or not obviously, but ViaCyte is another company that they have these packets that protect the cells for insulin replacement. And that's where he's leaving UBC to run that, but his lab's still staying at UBC, so something.
Okay, thank you. Thank you. There was another question over there.
Yeah, I was wondering, would you think it would be interesting to use the pathways that cancer uses to grow faster, to make the organs you grow quicker and have them faster by the patients?
Another very interesting question. So, do you think you can use cancer cell growth pathways to actually speed up the growth of cells in 3D printing?
Well, it's funny because we actually, the cancer cells that we grow in our 3D bioprinted tissues love our ink. They love being in 3D, and they are super invasive, which they aren't that invasive when you just grow them in 2D. So we don't tend to use that to speed up our differentiation process. But it is really interesting to see that cancer, like how differently the cancer behaves in a 3D printed tissue. We have a really interesting model where we printed healthy brain tissue in the presence of brain cancer tissue, and then we found a drug that would just selectively kill off the cancer while leaving the healthy tissue. And so that was one of the things we were really excited about, because we always talk about, we can use these for drug screening, and then we actually identified a novel drug using our tissues. But yeah, we haven't thought about that. I will always say that like cancer cells, because stem cells were discovered by cancer biologists through radiology and things like that. There's my friend who does cancer stem cell stuff, we always argue which is like the good stem cell versus the bad stem cell, because I always argue that like our pluripotent stem cells are the good ones, but he's like, but the cancer cell is the smart one because it's taking all the resources for itself. So I don't know if you have thoughts on cancer to share.
I actually did have a follow-up question. You kind of answered it already, but you did mention cardiovascular diseases, 3D printing, heart muscle perhaps. And you did mention GBM cells, glioblastoma cells. So what kind of research are you doing in cancer if you're not using the pathways to speed up the growth of cells?
Yeah, so we published a couple papers on glioblastoma using U87s. And as I said, we had this really interesting paper where we had the U87s co-cultured with healthy astrocytes. And because we wanted to show you could just kill off the tumors, because it's really easy to kill cells. But for cancer treatment, you just want to kill the cancer cells and not the healthy brain cells. So we just recently got in some cancer patient glioblastoma lines. It took me forever. Like every talk I gave, I was like, if you have any patient cells, please send them to us. And then my friend at UCLA was like, oh yeah, here you go. And so we've got the patient ones we're culturing now. It's a collaboration with a company called Zonula, and they have a molecule that targets these cell surface receptors that are highly expressed in the cancer cells. And they previously validated it in breast cancer. They read our paper. They were like, hey, can we send you some of this drug? We tested it, and we got some really, really interesting results.
We should talk about collaborations, by the way. Yes. I'm a medical physicist, so I'm interested in cancer research as well. Okay, I'm going to go to you first before I go back.
So I know that in organ transplants today, one of the big issues is the drugs that an individual would have to be on for an extended period of time to avoid rejection. So I'm wondering, I know it's years away, but I'm wondering if you have commentary, if we could 3D print an organ, such as a kidney, what would the impact be in terms of those rejection drugs that we currently have to use?
So the question was on organ transplant and really on the treatments following organ transplant, if we can make this an easier process on the body and perhaps avoid an extensive usage of drugs if we can use 3D printing.
Yeah, well, the real thing would be, because, as I said, you can do a blood draw and get stem cells made from your own cells. So they actually, in Japan, with these HIPSCs, they were doing this where they were making patient-specific eye tissues for treating blindness. And interestingly, what they found was it didn't actually need to be from the patient. You just needed to match on some of the major immune histocompatibility complexes in order to prevent rejection. And I will point out that that is in the eye, which is your CNS, so it is still a bit immune privileged. But in theory, you could make a patient, like you could take a patient's own blood cells, make them back into iPSCs, use those as the kidney, and then it would see you as self because they're your cells in that context. And so I think, and that's why iPSCs are such a powerful tool, is because it would potentially get around some of the, you would need to take immune suppressants because the tissue is actually made from your tissue. So yeah, I haven't seen, I just know with the eye stuff, and then it's, the CNS is a bit different because your immune system doesn't normally go there or see those cells, so it's a little bit easier to transplant into.
Yeah, it seems like a great idea for 3D bioprinting, but clearly it's not as straightforward, otherwise people would be doing it at this point. That's my take.
Well, I mean, we are printing people's tissues. It's just when you get into clinical trials, it's been, the field has been really interesting. I'm like, oh God, this is being live streamed. When Yamanaka won the Nobel, like Japan invested a lot of money into this technology, so Japan would be the leader. And so because of that, a lot of the stuff that they've taken to clinical trials in Japan to sort of beat everyone else, like normally, previously with stem cell stuff, like at conferences like that we attend, people would show their work and things like that, so then the field would know what was going on. But with Japan, because they wanted to be the leader, a lot of those processes, they just had to show the regulatory body and not the data to the rest of the world. And so that's why it was sort of interesting, because you only sort of hear about these things after the fact, where they were like, okay, so we were going to use patient-derived HIPSCs, but instead we are just using these other ones that are really similar to that patient's immune system markers. And so that's sort of where that is.
At least that's what we think where it is.
I think so, yeah. It's always like, oh, okay, yeah. You're just like, oh, and then like you see the results like 8 months later, and you're just like, oh, I wonder what failed or what didn't happen.
And we might never find out about these things as well, which is unfortunate, right? Because it would help the scientific community, but it's not always the case. Did we have a question here from you again, or no? Okay, okay. Let's go back to the nurse question. Thank you. So the question was if there are any trials using prophylaxis for HIV treatments, I suppose, using bioprinting.
Yeah, I'm not sure, because like the thing we've been more caught up on, I do know that people were using 3D printed lung for COVID testing with like the ACE receptor. There's like a lot of the organoid bioprinting stuff there. I haven't been as caught up on my HIV stuff recently. I just feel like, everyone's always like COVID. But it definitely is something that people have been using to sort of study the different viral binding. I'm trying to think what you would be, I guess you'd be looking at MSCs for HIV, like you'd be looking at the immune cells it would be binding to. I bet though, the company I was talking about who makes the vasculature, they actually have immune organoids, so they could probably screen on that, but they were using them for COVID, as we all did during, or whatever, everyone switched looking at COVID.
Because that's where the funding was?
I assume. Everyone, I felt like everyone in the biotech industry immediately was like flipped to making COVID organoids at the time. Oh yeah, no problem. Good questions.
Are there any other questions? Okay, thank you.
You mentioned Alzheimer's and Parkinson's. Does your work have any application in MS?
The question was if your work has any applications in multiple sclerosis.
We could use it to model it. We just haven't gotten down that disease pathology yet. We actually, although it's not announced, it looks like we're going to be moving back into like bioprinting for spinal cord injury. But there's a lot of things, like definitely MS would be a good one. The interesting thing about our systems with cancer is, so currently if you wanted to test a cancer drug to take it to clinic, you're usually implanting a human tumor in a mouse that lacks an immune system. And so the mouse is unhappy, it's an artificial system, et cetera. With our systems, you could actually spike in all human cells. So you could have both human immune cells in the presence like you would have in the actual body. There's also human immune cell therapies called CAR-Ts, so you can actually test your CAR-Ts in a fully human system. And so that's something we've actually talked a lot about with BC Cancer and Devika. We have been talking about, because you can actually have, for one of the first times, a fully humanized 3D system to test cancer therapeutics. So I'm pretty sure something similar would be possible with MS, because again, or I don't know if it's obvious, but our immune system plays a big role in modulating a lot of the different health states, especially like with MS, arthritis and things like that. So the fact that we could actually have human on human systems for some of these, I think is really exciting.
Go ahead.
You mentioned Japan's desire to be the leader. Could you comment on how we are positioned versus Israel, the States, the UK, Japan, in terms of our leadership or following?
So the question was on how Canada is positioned in terms of leadership in bioprinting compared to other countries such as Japan, US, UK, Israel.
So we do have a strong, I don't think you even give my standard talk, which is Canadians discovered stem cells. So Till and McCullough are Canadian. As you know, I'm American living here, but I like to tell everyone that because I think it's really important. So Canada does have a really big legacy. Neural stem cells were discovered by Canadians at Calgary. Peter Zanstra, huge stem cell researcher who's the head of the School of BME at UBC. So Canada does have a really strong legacy of doing really good stem cell work. Obviously, Peter Zanstra has founded Notch, which is taking over. The government does specifically fund a stem cell network that supports a group of researchers, including myself. So I think Canada sort of punches above its weight. And then also I found, as I was saying, like talking with Health Canada, they seem really open to using some of these new technologies. Politics aside, there was, down in the States, even just like I had to teach it in class, right? Because of the changes when under the Trump administration they're making to like how things were being regulated, which I'm not going to say that the FDA was doing like all the right things to start with. I'm not sure if less regulation was the way to go, but I could understand why some people would feel that way. So the states sort of went kind of wonky there, and it was more a lot of the preclinical screening stuff has really been led by pharmaceutical companies there because they just know it's more accurate at predicting what's going to be a therapeutic when it makes it to humans compared to the things you need in the regulatory pathway. Japan's really strong, and I know Europe has a pretty strong bioprinting presence as well, and they have some really interesting programs for funding through like their EU Horizon 2020 program, where they really encourage a lot of interplay. So, and I think in the Canadian government is like with the pandemic has coming up with a biomanufacturing strategy and realizing we needed to train more people and invest in this as well. So.
Thank you. One last question, I think, because we're coming up to the hour. Go ahead.
What about China and India?
The question is, what about China and India?
Oh, man. They, there's still lots of good research being there. I think it still is evolving. At this Australian conference where I was talking to some trainees from India, they're still, a lot of them are really interested in learning more about our bioinks and things like that, just because they are still using some more primitive technologies and things like that. China has some, it's just a really interesting place. They had the guy who CRISPR'd humans, and so although I think they're trying to crack down a bit on some of their regulations, I do know they were doing some stem cell. I know some people from some U.S. researchers actually were doing their clinical trials in China just because it was easier to get access, but also it's like the way their medicine's delivered is also a bit different. Like, as I said, I originally came from a spinal cord injury background, and so it was like if you were going to put in stem cell therapeutics into spinal cord injury, it was like you would have to recruit neurosurgeons because they treat spinal cord injury purely orthopedically, so it's kind of crazy. So so that's sort of I think a little bit on the take. Korea actually has a really strong bioprinting and additive manufacturing segment as well, so I know there's some really strong researchers there.
Okay, thank you very much. And since it is 7 o'clock, let's thank Stephanie for her fascinating talk, and thank you for coming.
Thanks, everybody. Yeah, and if you want to learn more, you can find my Google Scholar profile, and we have like a really adorable website for Axolotl if you want to see the bioprinters in action. And thanks again. It's always great to do science in person.